This video is going to cover genetic problems dealing with epistasis and incomplete dominance. So what we're going to start out with is the second page of the genetic problems that started with the rabbits, the solid in the spotted rabbits. And we actually did this problem already. And right here, I told you to write in the margin this word. Epistasis. And if you recall, epistasis involves some type of masking. So I actually already did the problem for you, so let's go to the GIMP board that I have it done, and then I'll go through it. So this is question number 2, dealing with epistasis. So genetic problems number 2, dihybrid. So this is a dihybrid, and we're going to show you an example of epistasis. So let's read what it says. If you have your handout, ready? In poultry, white, it's right up here, white, let me get another color here, may be the result of the homozygous state of the recessive factor O. See over here I have 2 little O's. Or it may be the result of the homozygous state of the recessive factor, those 2 little C's. You want to see that? And then it says color, the presence of both. See, both dominants are necessary for color. Then it tells us what the silky breeds and the white wanted dots are. So the silky breed over here has a formula, 2 little C's, 2 big O's. So what are they going to look like? They're going to be white. And then it tells us that the white wanted dots has a formula, homozygous dominant for C's, homozygous recessive for the O's. And they also are white because they have, over here, 2 little O's. And then it says, what would be the appearance of the F1s and the F2s in a cross between these 2 breeds? So if we, over here, made the silkies with the WWs over here, what will their offspring look like? Alright, so we have white parents. Both parents are white. Here's the F1s. This parent can only give big C, so all the offspring have to have has a big C. This parent over here can only give little C, so all the offspring, the F1s, have to be heterozygous for that first trait. Over here, this offspring over here, the silky, can only give big O's, so every offspring has to have a big O. This one can only give little O's, so they all have to be heterozygous for both traits. And then we need to find out the F2s. So we have to figure out what kind of gametes are possible. And over here, I did a FOIL method. Let me make that a little bigger so you can see it better. So FOIL first with first. Big C, big O. First with the last. So outer with outer. Here we go. So again, we're following FOIL. First with first, then the outers, then the inners, and then the last. That's so far we did first. That's smaller. Outers. Big C with big O. Inners. This one right here. Right here. And then the lasts. Last with last. Right here. Let's give you time to take a look at that. So what I want you to do is go ahead and turn this off and fill out the Punnett square. So go ahead and turn it off. And fill out your whole Punnett square. And now that you're back, we have to figure out what their phenotypes will be. So what are their phenotypes? In this case, there's actually only two different phenotypes, right? There's white and colored. So you have this phenotype and this phenotype. So over here we have this one and this one. Dominant one's always going to go first. So first what we're going to do is we're going to look through this, all the F2s, and look for all those that have at least one big C and one big O. And I did that, and I put a green check mark by them. And then I didn't really need to do this, but I went and looked for all those that had two little C's or two little O's. And I put a little orange circle there. And just so you can see the whole import at one time, made it a little smaller, so here you could see the whole thing. Now what we did, we had to go and count them now, and we found out there's 9 that are colored. Those are the ones with the green check marks. And 7 were white. 7 had 2 little C's or 2 little O's. On your test, you will not have to do an epistasis problem, but you will need to know the clue that this is epistasis. Look over here. When we cross two parents that are heterozygous for both traits, if you remember, we're always going to be expecting a 9 to 3 to 3 to 1 ratio with 4 different phenotypes. There are four different phenotypes in a 9 to 3 to 3 to 1 ratio. In this case, in our case of epistasis, we did not get the 9 to 3 to 3 to 1 ratio, we got 9 to 7. And we actually only had 2 different phenotypes. So not all epistasis is going to have a 9 to 7. This is just one type. But in all the different types, the clue will be that you do not have a 9 to 3 to 3 to 1 ratio. So what was this called? Epistasis. Let me make it over here. So this is epistasis. So there's some kind of masking going on. All right, let's go to incomplete dominance now. If you recall, this actually is a problem that we did when we were talking about complete dominance. I'm just putting it up here in case you want to take a picture of it. That word complete is kind of shaky. And I got a new mouse with my new computer. It's not quite as nice as my other one. This actually is complete dominance. This is our example with albino and big toes. And I'll make it a little smaller so you can take a picture of the whole thing if you like. All right, let me make it a little smaller. So this is our example that we did on our previous video with the albino, normal, big nose, no, big toes, small toes. Okay, that's that one. Now we're actually going to go to incomplete dominance. So this is the second page of our dihybrid genetic problems. The first page was the one with the normal, albino, big toes, small toes. And let's go take a look at this. This is a dihybrid. One of the traits is incomplete dominant, and one of the other trait is complete dominance. So let's go look at our clues. Well, let's look at our clues. I'll just put an I for incomplete dominance. That looks like a big T. Or incomplete dominance and complete dominance. So let's look for our clues. All right. Go ahead and look at it. Suppose that R1 equals red, R2 is white, heterozygote is an intermediate pink. So stop right there. We know that one has to be the incomplete dominant one. First of all, there are 3 phenotypes. Oh, catches and grabs a mouse pad. So 3 phenotypes. The way we're going to write it. Again, they do not always write it this way, and we'll do a problem where they're not writing it correctly. Where they're writing it with R1s, R2s. Can anybody think of any other clues? The clue is the heterozygote, and we would write the heterozygote R1, R2, which is going to be pink. Alright, so that's something in between. Our complete dominant well, we give us dominant is tall and short. We have 1, 2. Make sure you know these. How they're different. 2 phenotypes. How we're going to write again. This is not the best of clues because incomplete dominant does not always write it like that. But when they do, there's a clue that it is incomplete dominant. And so in this case, these both homozygous dominant and the heterozygote have the same trait. So the heterozygote has a dominant trait. So let's go ahead and cross these. I actually did this already for you, so let's go to that. And this time, we are going to be using the tree method or the branch method. So you can see how that works. So not always, but lots of times when you have a problem where you have an incomplete dominant and a complete dominant, lots of times the tree method is the best. So go ahead, let's go look at that. So on the top here, I wrote R1, R1 is red, R1, R2, pink, R2, R2, white. Our second trait, tall and short. Then you can see how I wrote that. And then they said that we're going to cross R1, R2, big T, little t, with the same. And so that's what I did. Now what I did, using the tree method, make this a little bit smaller for you. I did 2 monohybrid crosses. One with the R's, first trait, one with the second trait. So these ones in here. And these ones over here. Everybody see what I did? And then what I did was, I came up with the phenotypic ratios. Just the phenotypic ratios. The alphabetical order, so we're going to start out with this trait first. So notice we have, okay, we're looking at the phenotypic ratios. So these are red. These ones are red. And I might mess it up. This is red. These two are pink. That one's white. Look what I did. I make it a little smaller now so you can see it. There's 1/4 possible of red. Keep that as a 2/4. Don't simplify. 2/4 pink, 1/4 white. And our second trait only has 2 different phenotypes. So I made 2 branches. And then I went and looked at this 3/4 tall, 1/4 short. So 3/4 tall, 1/4 short. 3/4 tall, 1/4 short. And then we had to use our product rule, meaning we had to multiply. This could be 16 just so it didn't mess everything up. I didn't put the 16. But at the end, you have to add these all up, and they should equal 16. If it doesn't equal 16, then you did something wrong, because it should be 16 over 16, which would equal 1 or 100%. So I'll go 1 times 3, 3. 1 times 1, 1. 2 times 3, 6. 2 times, again, just look at the numerators, times 1 is 2. I'm multiplying. 1 times 3 is 3. 1 times 1 is 1. And I added them up. I did do it right. I did get 16. And these are the different phenotypes. Red tall, red small, pink tall, pink small, white tall, white small. So here it is. If you want to take a picture of it. Again, this was incomplete dominant. All right, incomplete dominant. The next problem on that same page, I'm not actually going to do it. Just going to point it out. Suppose that a horse is brown. All right, so you have brown. Notice they're giving us 2 letters. And then we have white, 2 colors. Heterozygote is an intermediate roan. Okay, so we know that that is incomplete dominant. First of all, they have 3 different phenotypes. 1, 2, 3 phenotypes. And roan The heterozygote is something in between. So if you don't know what roan is, something in between brown and white. All right, so we know that that one is incomplete. So the first trait right here is incomplete. So our first trait is incomplete dominance. Why? How can we tell? If I gave you a problem, make sure you can tell. Again, this one is not written with the R's, you know, the F1s, the R1s, things like that. F1 completely, look over here, furry, completely dominant over this. So we only have two, different color. We only have two phenotypes, furry and smooth. So what would the phenotypes of the following cross look like? So we could take a shortcut. So again, I'm not going to do that. Might want to try to do that, the tree method. This is the last problem for this video. And let's go take a look at this question because you might see something like it in the future. A rooster with gray feathers. Alright, so we have gray. Mated with a hen, which is gray. Alright, gray. Among their offspring, 15 chicks are gray. So 15 are gray. 6 are black. 8 are white. And if you remember from a previous problem, when we did a heterozygous cross, we had 1:2 to 1 ratio. Now, let's look at question A. What is the simplest explanation for the inheritance of these colors in chickens? And how are you going to figure it out? All right, we have gray with gray, and they have gray, black, and white. So, well, first of all, we know that there are 3 different phenotypes. And we know gray is something in between black and white. So what do you think is the simplest explanation for the inheritance? Is it complete dominance? Is it epistasis? Is it, what is it? What do you think it is? Well, I think it's incomplete dominance. Because we have 3 different phenotypes, and we have one that's in between the 2 extremes. Like blue and red, and you get purple. So if I made it to purples, I would get purple, red, and blue. Okay, so make sure you know how you figured it out that this was incomplete dominance. Then what offspring would you expect from the mating of a gray? Alright, we get a different color here. I'm just going to write over these words here. Alright, so we know that gray is going to be heterozygous, mated with a heterozygous, and we're not going to do the B one just to show you another way to do it. And these are the whites. So we're going to do a Punnett square. Here. And gray. Doesn't matter what side you put it on. Heterozygous. Black. Let's fill it out. Phenotypic ratios. Oop, yellow. So 50-50 chance. So we're going to have 50% are going to be black. And these are just the ratios of the possibilities. So we can say 2 black and 2 what? 2 gray. On the top of the page right here, there's some really good websites for genetic problems that show you how to do them, to give you little problems to do, to give you clues if you're not getting it right. They give you a lot of different genetic problems and a lot of practice. So it's a good one if you need any help. And that's going to be the last one for this video. The next video we're going to be doing problems with multiple alleles like blood type and hemophilia, so X-linked. So that's going to be our next one. So X-linked, color blindness and hemophilia, different dihybrids with that, including multiple alleles, blood type. Well, I hope you found this helpful.